Welcome everyone to today's seminar. This is the seminar that runs under the auspices of the PANGAIA project, which is project number 872539, funded by the EU. And its aim is to fill the gap between the rapidly increasing power of DNA sequencing technologies and the current limitations in resolving the inherent complexity of the forthcoming volumes of data. So first, let me ask Paola Bonizzoni to introduce today's speaker.
Thanks, Thomas. Welcome to everybody to this seminar series. You're welcome. So it's a nice pleasure to meet you here. And it's my pleasure to introduce Simone, since I know him from his bachelor degree, so he is just a friend, and I am very happy that he's going to give this talk today. Simone Zaccaria is currently the group leader of the Computational Cancer Genomics Research Group at the Cancer Institute in London. He's also visiting scientist at the Francis Crick Institute, and moreover, he serves as lead of the informatics core of the Cook Lung Cancer Center of Excellence. Simone spent 4 years in the US working with Professor Ben Raphael, as a postdoctoral research associate, and he received his PhD in computer science at our University of Milano Bicocca under my supervision. So I'm very happy to invite him today to talk about investigating the role of somatic copy number alterations in shaping the landscape of cancer genomes. Let me remind that Simone is currently mainly involved in the development of computational methods to investigate cancer evolutionary processes from next generation sequencing data, cutting-edge sequence technologies, of course, including single cell and multi-site sequencing. So welcome, Simone. I just invite you to start. Thanks again for accepting our invitation.
So first of all, I would really like to thank all of you for inviting me here today. And in particular, today, I would like to tell you about some recent works that we have developed to investigate these large genomic alterations or structural rearrangements that characterize the landscape of cancer genomes. And before start, I would like to mention that most of the works that I will present today have been developed at Princeton University, where I was a postdoc, and then I'm continuing to work on these studies at my new position in my new research lab at the UCL Cancer Institute. So let's start from the beginning. We know very well that cancer arises from a complex evolutionary process in which somatic mutations accumulate over the time and in different cells. And this complex evolutionary process always leads to very heterogeneous tumors, such that a tumor, as you can see here in the figure, is composed of distinct subpopulations of cells, also called clones, where each clone is characterized by a unique complement of somatic mutations. And so, due to the complexity of this process, there are typically 2 very key questions that we aim to answer. The first one is to quantify this intra-tumour heterogeneity, meaning that we aim to identify the distinct clones that are present in a tumor, to quantify the prevalence of these clones, and to identify the somatic mutations that characterize each of these clones. And then, based on these answers, the second important key question is actually to aim to reconstruct a tumor evolution, meaning that we aim to reconstruct a phylogenetic tree that describes the ancestral relationship between the clones that we identified and reconstruct the corresponding time ordering of the mutations. we know that answering both these questions actually have a critical impact on both clinical and biological applications. In fact, by reconstructing the tumor evolution, we are able to identify mutations that drive cancer progression. And so these clearly enable to identify potential drug targets, but also it allows us to reconstruct the complex metastatic migration patterns that correspond to the dissemination patterns that lead metastatic cell to migrate from a primary tumor to other distant metastases. So answering these questions is extremely important, and in order to start to address these questions, we really need to go back to the main subject. And the main subject are clearly somatic mutations. In fact, we know that there are several different kinds of somatic mutations that can occur and affect the genome of cancer cells, ranging from mutations that affect a single nucleotide as a single nucleotide variants, SNVs for short, to mutations that affect much larger portions of the genome. And today I would like to specifically focus the attention on one of these that are called copy number alterations. These are somatic mutations that affect large genomic regions, whole chromosomes, and even the entire genome with whole genome duplications by amplifying or deleting the number of copies of these large genomic regions. So here in the slide, we can find an example where we have a normal cells with 2 copies of one chromosome. Due to an amplification, we can end up having 3 copies of these chromosomes, or due to a deletion, we can end up having only one copy of that chromosome. And so since the changes that are provided by these copy number alterations can be actually particularly substantial to the structure of the genome, we may assume that these changes are actually not very common. However, this is not the case. In fact, when we went to analyze thousands of individual tumors across international cancer consortia, we actually found out that these copy number alterations are extremely prevalent and extremely frequent across nearly all cancer types. We can see an example of these results here in the slide, where in this heat map, every row corresponds to a different tumor that are classified according to the different cancer types, and then the columns represent the entire genome across all chromosomes. And so we represent in white genomic regions that are supposed to be normal diploid regions, and then in either red or blue we represent amplifications and deletions. And so you can immediately see from this heat map that we can really barely find any tumor that is completely white, and nearly all tumors are really substantially affected by many copy number alterations. And so these copy number alterations are extremely pervasive across cancer. And for this reason, they represent a fundamental marker that now we can try to use to answer the 2 key questions that I proposed at the beginning. So the important question now is, how can we identify these copy number alterations? The standard approach that is adopted by nearly all cancer sequencing studies is using aligned DNA sequencing grids that are obtained from a bulk tumor sample. And in particular, in order to give you the intuition on how we can use the number of sequencing grids that are obtained across the entire genome to identify these copy numbers, I have a little toy example to show you. Imagine that you have a fruit puzzle that is composed of 2 yellow pears and 2 red apples. Now, according to the sequencing process, let's assume that we scramble up these puzzles, so we don't really have the complete picture, but instead we only find out all the different pieces of this puzzle. So from here, we cannot directly observe the number of pears and apples that we had in the puzzle, but we can only observe the number of pieces with different colors. So if we want to answer the question on how many fruits were present in my puzzle, what we can do, and probably you have already guessed, we can try to count the number of red pieces, compare it with the number of yellow pieces, and in this case we can see that pretty much we have the same number of red and yellow pieces of the puzzle, so we can guess that we have the same number of pears and the same number of apples. However, if I now show you this different situation, you can see in this different puzzle that the situation seems like pretty different. We see that there is a much higher number of yellow pieces, but a very low number of red pieces of the puzzle. And so in this case, we may guess that actually we expect to have more pairs than apples. This is in fact the originating puzzle of this study example. So this is actually the same intuition that underlying the basics of these copy number analysis. So we go across the entire genome and we count the number of sequencing reads that are obtained from the different regions, and we can then translate this number of sequencing reads into the different copy numbers. So this seems actually like a naive approach that we can apply. But unfortunately, it's not so easy. It's not so easy because there are several challenges and issues that make this problem particularly complicated. And one of the major problems is actually the presence of subclonal copy number aberrations. What are these? So since we are starting from a bulk tumor sample that is a mixture of millions of different cells, the cells that we are sequencing can also be affected by different copy numbers. They can have different copy numbers for the same genomic region. This means that for some genomic regions where there are these different copy numbers, we will not see a number of sequencing reads that we can directly translate into an integer number, but we will rather see some intermediate state. So we are not really sequencing only one puzzle. We are sequencing multiple puzzles that are mixed together in unknown proportions. And the challenge in this case is to try to take apart the different components, so to identify the different copy numbers that are present jointly in our sequencing reads and the corresponding proportion. So it's really a factorization problem. And this factorization problem is extremely complex. Despite the complexity, however, we said how important it is to infer these copy numbers. And so for this reason, there is a very large literature of methods that have been proposed to attempt to overcome the challenges of these problems. And in order to do that, the approach that is applied by these methods is to try to introduce restrictive and simplifying assumptions that make the problem slightly more accessible, and then we can solve it. However, nowadays, there is actually another approach, another alternative approach that we can attempt to follow. In fact, there are new sequencing technologies and new sequencing approaches nowadays that actually provide additional sources of information. And so the question that we want to propose today is whether we can use these additional informations in order to improve the inference of these copy number aberrations and the inference of in general, the tumor clonal composition. One example of these new sequencing approaches is, for example, the possibility to obtain multiple samples from the same tumor. In fact, when we obtain multiple samples from the same tumor, we are sampling cells from the same tumor that share a same common evolutionary scenario. And so the question is whether we can leverage the joint information across multiple samples to now improve the inference of copy numbers. A second example that I will not tell you today about, but that is very important for me, is actually corresponding to even more revolutionary and cutting-edge sequencing technologies that are single-cell sequencing technologies. In fact, there are very recent technologies that nowadays are enable us to sequence the whole genome of thousands of individual cells in parallel. So these represent a sort of dream for the analysis of intra-tumour heterogeneity, but the main drawback of this data is that actually we obtain a very limited amount of data per cell. So that even in this case, we have hard challenges that we need to overcome. And the question again is whether we can leverage the joint information across all the cells to overcome these challenges. So the underlying theme in my research group is really trying to answer the question of whether we can introduce computational methods to bridge this gap, whether we can design algorithms that leverage the joint information across multiple samples that we can obtain from these fascinating technologies in order to improve the inference of the clonal composition and obtain novel insights about tumor evolution. And today I will tell you about two very specific examples in this topic. The first one, that is actually a method that we recently introduced to identify these large copy number alterations jointly across multiple bulk tumor samples obtained from the same patient. And then I will tell you about an even more recent work in which we aimed to combine these copy number alterations that we can infer with standard analysis of SNVs in order to reconstruct the complete evolution of the tumor. So let's start from the first part. That is about the possibility to obtain multiple samples from the same tumor. In fact, these samples nowadays can be obtained in multiple forms. For example, they can be obtained from multi-region samples, from longitudinal samples that are obtained at different time points, or from the sequencing of primary tumor and matched metastases from the same sample. And actually, something very important to say is that this idea of jointly analyzing multiple samples is not something new, but it's an idea that has already been demonstrated to be extremely important and extremely powerful in the analysis of SNVs, of single nucleotide variants. In fact, maybe some of you have already came across these two-dimensional plots that typically are used to represent the frequency of mutations in two samples obtained from the same patient. So every axis corresponds to the frequency in one sample. So this is one sample, this is a second sample that is obtained from the same patient, and we can analyze the mutations by jointly considering the frequency in these two samples, and we can identify clusters of mutations. So several studies have demonstrated the power of this kind of analysis. But very surprisingly, there is actually no comprehensive method nowadays that enable us to do the same for copy number alterations. So in order to overcome this limitation, we recently introduced Hatchet. Hatchet is the first comprehensive method that enables us to identify allele-specific copy number aberrations and also whole genome duplications jointly from multiple samples obtained from the same patient. And today I will tell you about one of the key underlying ideas of Hatchet that is related to how we can identify different copy number states from the DNA sequencing data itself. And I will let you discover the other key innovations in the recent related publication. So the standard approach that is adopted to use DNA sequencing data to identify the different copy number states is called the local segmentation. And the idea is actually pretty simple. So what we do in these standard methods is actually to consider a first important signal that we obtain from DNA sequencing data that is called the read-def ratio, and that is simply corresponding to the number of sequencing reads that we observe across the genome. So the same concept that I told you before. So by tracking the changes in the number of sequencing reads that we observe, we can identify the presence of large alterations. For example, we can see that chromosome 3 here has been amplified, while chromosome 13 has been deleted, and so we only have one resulting copy. However, this is actually not the only signal from DNA sequencing data that we use, but there is also a second signal that is actually leveraging germline heterozygous SNPs that characterize the human genome. In fact, when we have a heterozygous germline SNP in a normal diploid chromosome, we actually expect that the frequency of the two nucleotides to be actually pretty much the same. So we expect the frequency of the nucleotides to be 50-50. However, due to the occurrence of copy number alterations, this frequency can substantially change. For example, if one of the two chromosomes get duplicated, now we have two copies of the nucleotide C, and so we expect the frequency of the nucleotide C to be much higher than the frequency of nucleotide A. So this is a second signal from DNA sequencing data that we can use. It's called the B allele frequency, and we can also compute this signal across the entire genome. And moreover, this signal also allows us to identify these allele-specific copy numbers, so splitting the total number of copies that we have specifically for the two different alleles that are contained in our genome. So given these two data, the standard idea of local segmentation to identify these states is actually pretty simple, and it is to march along the entire genome and to combine any pair of consecutive genomic regions that have the same values of these two signals. So in this way, we can create these stretches, these long genomic segments that represent the effect of these copy number alterations. However, if we now look across the entire genome, actually these most of copy number alterations will result in genomic regions that typically occupy only few distinct copy number states. In this specific tumor, we can see, in fact, that there are only 3 states, 1 or 4, maybe if we also consider BLE frequency. But we have a state of genomic regions that are amplified, so that have 3 copies. We have a state of all the genomic regions that are not aberrant, and then we have a state of genomic regions that are deleted here below. So the first idea that we introduced is that since we have this consistency and this limited number of copy number state, what we can do is probably to leverage the joint signal across the entire genome by pulling together all the genomic regions together. In fact, if we pull together all these genomic regions, we forget about their genomic location, we can see that, as we expected, the genomic regions really form few distinct clusters, a few distinct groups of genomic regions that are described by the 2 signals that I showed you below. And in fact, these different groups of genomic regions actually correspond to the different copy number states that we were expecting. So we have the non-aberrant genomic regions down here, we have genomic regions that are amplified here to the right, and then we have genomic regions that have been deleted here on the top. So by pulling together all these regions, now we have the power to really better distinguish what are real variations versus standard variants of our DNA sequencing data. But there is actually a second very important reason for this approach. And the second, even more important reason, is that actually we can very easily extend this approach to work simultaneously across multiple samples. In fact, we can cluster genomic regions, we can color in the same way the same genomic regions across all the samples, and we can now leverage the information across all the samples to identify these clusters and identify possible changes. For example, we see this red cluster that actually corresponds to chromosome 13 that is here on the top in all the samples. So these seem to appear like a clonal copy number aberrations present in all tumor cells. But we can also very easily identify large genomic regions, for example, these dark green, that actually completely and very substantially change their position across different samples. And so these would indicate the presence of very clear subclonal copy number aberrations. So by putting together this innovation with few other innovations, we really wanted to assess whether we can improve the performance of inferring copy numbers. But unfortunately, simulating actual DNA sequencing reads in the case of copy number alterations is not easy. It's not easy because these copy number alterations substantially change also the length of the genome of the different tumor clones. So we need to be very careful in treating sequencing reads that come from genome of different lengths. And so to do this, we introduce Mascot, that is a method that uses previous standard methods for simulate sequencing reads and adapt these methods with appropriate corrections for the different genome lengths in the case of copy number alterations. So we simulate a large number of BAM files for 256 tumor samples, and we compared Hatchet with previous current state-of-the-art methods. We found out that Hatchet was actually able to clearly outperform all the other methods when considering multiple samples jointly, but even when considering a single sample. Meaning that not only the joint analysis of multiple samples, but even the other innovations actually played a very important role. So in order to assess what is actually the biological impact of having like these more accurate analysis, we decided to analyze 2 different cancer sequencing datasets. One characterized by 10 metastatic prostate cancer patients with 49 samples, and one characterized by 4 metastatic pancreatic cancer patients with 35 samples. And we actually decided to consider these 2 specific datasets because previous analysis of these 2 datasets actually reached completely opposite conclusions about the reconstruction of tumor evolution. In fact, the previous analysis of the prostate dataset identified the crazy presence of subclonal copy number aberrations in all the samples, and this led to identify very complex polyclonal migrations across the different metastases. Instead, the second study didn't identify any subclonal copy number aberration on the pancreas, and this resulted in identifying very simple monoclonal metastatic migrations. So we wondered whether the differences between these two analyses were really, capturing some important cancer type difference in the two patients, or it was simply related to some technical and bioinformatic difference. So we applied Hatchett on these datasets, and we indeed found that Hatchett actually identified substantially different solutions than the previous analyses that were more accurate, but also completely changed the downstream analysis. And I will show you two examples about this now. First, on the pancreas dataset. So in the pancreas dataset, the previous studies excluded the presence of subclonal copy number aberrations, but when we analyzed with Hatchett, we very clearly found a substantial presence of subclonal copy number aberrations. Here in the slide, you can see an example of this in one sample, where this light green cluster really occupy an intermediate position between the states with 2 number of copies and the state with 3 number of copies. So this is a very clear subclonal copy number aberration. And very interestingly, actually this cluster corresponds to the entire chromosome 4. Moreover, when we jointly analyze all the samples from the same patient, we actually found out that the 2 clones that were present in this specific sample were separately present into other 2 samples in liver metastases. In particular, we found out that this lymph node 1 contained 2 distinct clones, one green and one blue, such that the green clone was also present in the first liver metastasis, while the blue clone was present in the second liver metastasis. And so these actually revealed that probably the metastatic migration patterns are much more complicated than what we expected from previous analysis. I will now show you a very quick example also on the prostate cancer dataset. So in this case, previous analysis used the complex subclonal copy number aberrations that were pervasively identified in all the samples to analyze the single nucleotide variants. And in particular, again, they used these 2-dimensional plots to jointly analyze the frequency of the mutations in 2 distinct samples. And they identified this large cluster of mutations, of blue mutations, that actually had a very low frequency jointly in 2 samples. And this was provided as a very strong evidence for the occurrence of complex polyclonal migrations. However, when we applied the same analysis using the clonal copy number aberrations inferred by Hatchett that were equally plausible by the data, we actually showed that the frequency of the same mutations completely changed. The mutations are now incorporated into the major cluster, and these actually tell a much simpler story and indicate much simpler monoclonal metastatic migrations. So using Hatchett, we showed that actually the accuracy of identifying copy number alterations is actually very important and very strongly impact the downstream analysis of tumor evolution. We also decided very recently to apply Hatchett on a large pan-cancer dataset with more than 250 tumors. And we also found out that Hatchett was able to reveal the pervasive presence of whole genome duplications. Whole genome duplications are actually these very large genomic alterations in which the entire genome of a cancer cell completely duplicates. And we actually don't know how much is the prevalence of these events in the tumor, but actually Hatchett revealed that it should be pretty high, inconsistent with other recent studies. And in particular, you can see here in the top half of the heat map that most of the tumors have a gray color that correspond to diploid, but instead the tumors on the bottom half have extremely and much higher colors that correspond to much higher copy numbers. And these in fact correspond to the identification of whole genome duplication. And moreover, even more importantly, we actually found out that this difference was actually strongly correlated with the deletion of the gene TP53 in chromosome 17 that is actually known to be related with high genomic instability. So to conclude, I told you about Hatchet, that is the first method that introduced a framework that enables us to jointly analyze multiple samples in order to infer copy number alterations. I showed you that these allow Hatchet to outperform previous methods and also to infer more accurate copy number alterations in cancer sequencing data, resulting in more accurate and important insights into the reconstruction of tumor evolutions. Now, I would like to continue with the second part. And in this second part, the question, the underlying question, is whether actually we can use the copy number alterations that now we can infer with Hatchet in combination with standard analysis of SNVs in order to better characterize the evolution, the tumor evolution. And so let's do a quick step back. So as we said at the beginning, cancer arises from an evolutionary process that starts from normal cells. So a normal cell accumulates a certain initial set of somatic mutations. These somatic mutations confer some fitness advantage that will lead the cells to undergo a clonal expansion. And so we have the formation of the first clone. And then this process is repeated multiple times throughout the accumulation of new somatic mutations that lead to further clonal expansions. And so what we do in our field is actually to obtain a sample or multiple samples of this tumor from a time point. And at this time point, our data will belong to different tumor clones. And the goal actually in reconstructing tumor evolution is actually to answer the question of whether we can reconstruct the ancestral relationship between the different clones that are present in these data. And we typically do this by using a phylogenetic tree in which every vertex corresponds to a different tumor clone. And then every edge is labeled with the corresponding mutations that occurred in the ancestral transition. Very importantly, actually, every edge is not typically characterized by a single mutation, but is rather characterized by a mutation cluster, a group of multiple mutations. And there are actually two very important reasons of why this is happening. The first one is biological. And the biological reason is that actually the first cell that is the founder of any clone will actually contain multiple unique somatic mutations. And all the somatic mutations that are present in the first cell of a distinct tumor clone will also be inherited by all the descendant cells in that clone. And so all these mutations that occurred in that cell will also be present in all the descending cells. The second one is actually related to the limited resolution that we have in DNA sequencing data. In fact, we don't sequence an infinite number of reads. And therefore, according to the different sequencing reads, to the different number of sequencing reads that we obtain, we might have different powers to distinguish mutations with very similar frequencies. And so sometimes these mutations with slightly different frequencies are just incorporated into the same clone. So identifying mutation clusters is extremely important. We expect to have these mutation clusters there. And if we are able to identify these mutation clusters, we can attempt to reconstruct the corresponding tumor evolution. So the question now is, how can we identify these mutation clusters? So the standard approach that is adopted by nearly all cancer sequencing studies is actually to try to identify mutation clusters as mutations that are present in the same proportion of cancer cells. So we assume that if two mutations are present in the same proportion of the cancer cells, then they must be present in the same mutation cluster. And this quantification, the quantification of this proportion, is actually done using a metric that is very famous, very well known, that is called cancer cell fraction, or CCF for short. So mutations that have the same CCF, like the moon and the square mutation in this example, will be identified as mutations present in the same mutation cluster. Unfortunately, we actually do not observe these CCFs. Instead, we observe sequencing data, and from sequencing data, we can compute something different, that is the variant allele frequency. So we can compute the frequency of reads that contain a certain mutation. So very early approaches actually relied on the assumption that mutations that have the same CCF will also have the same variant allele frequency. And so the idea was simply to cluster the mutations according to their values of variant allele frequency. So we identify groups of mutations that have pretty much the same values of variant allele frequency, and these represented our mutation clusters. And several methods have been developed to do this. Unfortunately, we know nowadays that this assumption was wrong, and this is wrong due to the presence of copy number alterations. In fact, copy number alterations can actually affect the number of mutated copies for a single SNVs within the cells. They can, for example, amplify the multiplicity of the mutation. We have an example here in the slide, where two mutations belonging to the same cluster are jointly introduced into the same clone, and then the square mutation is further duplicated and amplified due to a copy number alteration in the second tumor clone. So now we have two copies of the square mutation, but only one copy of the moon, and therefore we expect that the frequency of this square mutation will be higher than the other mutation. So we cannot really identify these two mutations as being part of the same mutation cluster. So in order to overcome this challenge, more recent and several methods have been introduced with the idea of trying to correct this variant allele frequency for the presence of copy number alterations. So the idea is that starting from the variant allele frequency and the copy number alterations that can be inferred independently, for example by Hatchet, as I told you in the first part of the talk, we can attempt to estimate the CCF of the mutations. And now that we have the CCF of the mutations, we can cluster the mutations together and identify these mutation clusters that will be used to reconstruct the evolution of the tumors. Unfortunately, even this approach has two important limitations and issues. The first one is that actually this inference of CCF is not straightforward, but it is extremely complicated due to the presence of multiple equally plausible solutions. So we have generally uncertainty about the values of CCF that can be inferred. Here in the slide, you can see an example for a single mutation with certain copy numbers. We can infer two different values of CCF that are both equally plausible. So in order to overcome this challenge, these existing methods typically apply some simplifying assumptions. For example, one of the most common assumptions is called CMM, that stands for constant mutation multiplicity. And it means that it assumes that all the tumor clones must have the same number of mutated copies for every mutation. However, this is clearly a non-realistic assumption in the field, and so the problem is whether this is actually tossing away solutions that could be necessary to reconstruct accurate tumor evolution. There is also a second and even potentially more important limitation. That is actually that copy number alterations cannot only result in amplifying the number of copies of one deletion, but they can also delete mutations and they can result in mutation losses. For example, here in the slide, we have again the same two mutations in the same cluster, but now the square mutation is not amplified, but it is deleted due to a deletion, a copy number deletion. And due to the result of this deletion, we completely lose now the copies of these square mutations. So the problem is that since the mutation gets completely lost, also the CCF of the two mutations will be different. So the proportion of cancer cells that have at least one copy of the mutations will be different in the two cases. And so we cannot identify that the two mutations are part of the same clusters due to the presence of these different values from the CCF. So to summarize, there are really two issues that are undergoing currently. The first one is the uncertainty that we have in the estimation of these CCFs. And the second one is that mutations that are present, that are part of the same cluster, might have different values of CCF. And so the CCF cannot really be used to identify mutations from the same cluster. There are actually two classes of methods that have been currently proposed to face these challenges. One that is typically based on mutation clustering, So these can deal with very large number of mutations, but they use unrealistic assumptions to get rid of this uncertainty, and also they use a CCF, so they do not account for mutation losses. There is also a second class of methods that actually try to take into account the presence of mutation losses, and they do this by trying to reconstruct the complete tumor evolution for all the mutations and for all the copy numbers jointly. But this typically results in extremely complex evolutionary models that do not scale to the realistic numbers of mutations and samples that we might have in existing cancer sequencing data. So in order to try to face both these issues, we introduced recently the DCF study. The Decipher study. And in the Decipher studies, we really try to introduce two key contributions that aim to overcome these challenges that I've just mentioned. The first one, in order to deal with mutation losses, we actually introduce a new statistic, DCF, that is an alternative to the CCF, to the standard CCF, but enables to accurately correct for mutation losses. And the second one is to deal with uncertainty. And in order to do that, we introduce a simultaneous clustering across all the mutations and across multiple samples without the use of unrealistic assumptions. And I will tell you some details about these 2 points now. First, the DCF that stands for Descendent Cell Fraction. So while the CCF is actually defined as the proportion of cancer cells that have at least one copy of the mutation, the DCF is defined as the proportion of cells that descended from a mutation, from a clone in which the mutation occurred. So these cells, the second tumor clone will be accounted by the DCF of this second mutation because it descended from a tumor clone that actually acquired the mutation. And so thanks to this difference, now the DCF is able to take into account potential mutation losses. And so mutations that are present in the same cluster will now have the same values of DCF. And so DCF can be used as a proxy to infer these mutation clusters. Moreover, we showed very importantly that actually the DCF of every mutation can be computed by analyzing independently the evolution of every mutation. In particular, we introduced this small phylogenetic tree that is called the state tree that only describes the evolution of each mutation independently. You have two examples of two trees here. And we show that these state trees with the corresponding proportions are actually sufficient to compute the corresponding DCF. And this is a very important result because actually we demonstrated that all the state trees for one specific mutations can actually be enumerated very efficiently. And even more importantly, we demonstrated that the proportions related to each state tree can be obtained uniquely. So this gives us the possibility to compute the DCF using the corresponding state tree for a single mutation. But again, we still have the trouble of uncertainty. In fact, as well as for CCF, even in the case of DCF, we can still result in inferring multiple equally plausible values of DCF. Here in the example, you see two values of DCF for the same mutation. However, we actually reason that different values of DCF actually will correspond to the presence of different clusters. In particular, the number of different values of DCF will relate to the different number of mutation clusters that we have, and therefore to the inference of different number of tumor clones. So under a principle of parsimony, we can actually try to minimize this number of clones that are present in a cluster. And we can do that actually if we consider multiple mutations simultaneously. Because if we consider multiple mutations simultaneously and we have multiple options for these mutations, we can actually try to choose the options that minimize the number of different values of DCF. And therefore, we result in minimizing the number of clones present in our tumor under a principle of parsimony. So this results in a complex problem that simultaneously compute the DCF and cluster the mutations. And in order to solve this complex resulting problem, we introduced the Decipher algorithm. So the Decipher algorithms aims to solve this complex clustering and estimation of DCF using a coordinated descent algorithm that split the complex problem into two subproblems. One that aims to identify the DCF of the different clusters. So what are the underlying centroids of the different clusters? And the second subproblem that attempts to assign every mutation to the most likely cluster. And we do this by using a probabilistic model that is based directly on the observed sequencing reads. So by alternatingly solve these two subproblems, Decipher will be able to solve also the more general problem. And in order to overcome and to deal with the potential presence of local optima, we repeat this process using a high number of random restarts. So thanks to this algorithm, starting from the read counts of the mutations that we can observe from sequencing data and the copy numbers and proportions that can be inferred by algorithms like Hatchet, we can now identify mutation clusters and the corresponding values of DCFs that can be used to reconstruct the tumor evolution. So we assess the performance of Decipher by comparing it with the most common algorithm for mutation clustering, that is PyClone, and we actually observe that Decipher significantly improves recall without loss of precision and by more accurately inferring the number of clusters, and therefore more accurately inferring the different number of clones that we have in our tumor. Again, in order to really evaluate what is the biological impact of these important innovations, we considered the same prostate dataset that I showed you before. And we observed that actually Decipher was resulting in correcting the mutation clusters for a very substantial number of mutations across all the patients. So here you see the average number of mutations for which a correction has been applied with different types of corrections across all the patients. And in particularly, we observed that for some of these patients, the corrections were actually extremely important, and they were resulting in very different reconstructions of tumor evolutions. I will show you two very quick examples to demonstrate this. The first one is in patient A17, and for simplicity now, I will just describe this example for 300 mutations in chromosome 5q. So here we have the variant allele frequency that are computed for these 300 mutations across two samples. And when we applied existing methods to this situation, we actually observed that these existing methods identify the presence of two distinct clusters, one brown and one green. And this actually resulted in the reconstruction of a very unrealistic tumor evolution. In particular, this reconstruction of tumor evolution was particularly unrealistic because we identify homoplasy for 142 mutations, for half of the mutations. So homoplasy corresponded to the occurrence of the same mutations in both the alleles of the same genomic position. It is considered to be an event that only rarely occurs during tumor evolution, and in this case, we would have like this rare event occurring for half of all the mutations occurred in chromosome 5. So this seems to be particularly unrealistic. However, when we applied Decipher on the same dataset, we actually found out that Decipher was collapsing these two clusters together into a unique cluster. And this resulted in the reconstruction of a much simpler tumor evolution. And this was thanks to the fact that Decipher does not rely on the unrealistic CMM assumption, and it was able to identify the presence of the same mutation with different multiplicities in two distinct tumor clones. So one copy in the first tumor clone and two copies in the second tumor clone. An even more interesting example, we actually found it in patient A12. So again, for simplicity, I'm just focusing here on the 100 mutations in chromosome 6q. In this case, when we applied existing methods to this situation, again, we observed that existing methods identified the presence of two distinct clusters, one pink and one green. And this resulted in a very complex reconstruction of tumor evolution with 4 clones. However, when we applied the Decipher on the same data, we observed that, again, all the mutations were actually collapsed into the same mutation cluster, and this resulted in a much simpler reconstruction of tumor evolution. And again, this was possible because the cipher is able to identify that all the magenta mutations that were present in the black allele has been deleted. And so here we had a mutation loss that was resulting from the loss of the entire black allele. And this was actually strongly supported by the orthogonal copy number analysis, because we indeed showed that the variations in the frequency of the magenta mutations were really just tracking the frequency of the deletions in the different samples. So with this, I would like to conclude also the second part. So I hope that I convince you on how these simplifying assumptions that have been widely used in literature and the fact that we do not account for mutation losses represent a strong limitation for reconstructing tumor evolution. So we introduced a new statistic that is able to account for mutation losses and a new algorithm that simultaneously compute this DCF and perform a mutation clustering in order without the presence of unrealistic assumptions. And we showed that the cipher outperforms previous methods and also result in much more realistic reconstructions of tumor evolution. So with this, I would really like to conclude, and I think that this cartoon actually well reflects the contributions and the work that is undergoing in my research lab. So starting from the data that are produced by very cutting-edge sequencing technologies, we try to design computational methods that are able to leverage the joint information across multiple samples in order to infer the tumor clonal composition. And then we design methods that try to reconstruct the tumor evolution. So with this, I would like to conclude. There are many people that I have to thank here, but first of all, I would like to thank all of you for the attention, and I would be very happy to answer all your questions.
Okay, thank you very much. So are there any questions? So Tomáš, please, Semeš.
Oh, hi, Simone. Thank you very much for your very interesting talk. So we are also trying to do CNV analysis, but we are focusing on circulating cell-free DNA, which brings in some more challenges. But I see that as a problem that might be dealt with your hatchet lineage of research. But I would like to ask regarding your algorithms. Did you consider also that cancer cell can also be of different ploidy than a diploid? Because in most of your slides it was shown as diploid, but there are certainly different tumor types that in which triploidy cells are involved, or even cells that cannot be put into the same bunch of or same package regarding its even ploidy, because they can have really uneven counts of different chromosomes in them. And this can be striking in certain types of tumor, and that may bring some complication when interpreting your data.
Yeah, well, this is a great question, but actually really one of the main innovations of Hatchet was really the possibility to properly treat changes in ploidy, especially due to the presence of whole genome doublings. And can I actually share the slides back? Sorry.
Sure.
Can you see my slides? So I didn't discuss about the algorithmic innovations that we introduced to deal with changes in ploidy, but I showed you this result that is actually, a recent result from this pan-cancer analysis. And you can see that actually the cells, all the cells, all the tumors that are in the second half actually correspond really to the case that you were mentioning. So these are all cells that underwent substantial changes in aneuploidy. So, and in this case, it was identified whole genome doubling, the change in these ploidy. So here we can have, mostly triploid cells that are the yellowish cells. So we can have tetraploid cells that are the orangey cells. And we can also have actually cells that still appear like diploid, but just because they underwent a lot of losses after they accumulated the whole genome doubling. So the answer is actually yes. And the idea is that the same clustering that I showed you before, so this new structure that now we can capture across multiple samples, give us really the power to distinguish whether the tumor clones is characterized by subclones or changes in ploidy. And actually we also, in the paper, we argue that these decision cannot be made from a single sample because changes in the clustering due to either subclonal aberrations or whole genome duplications or aneuploidy, they can look like the same in the data. And it's only when you look across multiple samples that you're really able to distinguish whether subclones were present or it was really due to the presence of a whole genome doubling.
Okay, thank you. And did you have a chance to also look at some circulating nucleic acids data from cancer patients?
This is also actually a fantastic question, and we are actually working right now also with few collaborations in analyzing the circulating tumor DNA. And in particular, the main advantage that we can use here is really the analysis of multiple samples. So like in the first analysis, we showed that since you are analyzing multiple samples, what you can do is actually to take the information that you have from high purity samples and use these samples, this information to better analyze tumor samples with low tumor purity. For example, you see that this sample over here, this is like the tumor purity, it's only the 28%. The data are all mixed together, but we are still able to separate them very well because we are actually incorporating and leverage the information that we have at a much higher resolution from other samples. So this is actually, when you have both ctDNA samples and samples from a primary tumor, an approach that is pretty powerful that we are trying to test. And we are also testing, the same approach when only analyzing ctDNA samples without prior information. But in order to do that, you really need to increase the power of your signals in DNA sequencing data. And we believe that the best way to do it is actually to incorporate the phasing of germline heterozygous SNPs. And this is something that is done, for example, by some previous methods like Battenberg. And so we are also incorporating now phasing inside Hatchet so that we can go to much lower resolution samples, even without prior knowledge. That is, for example, the case of ctDNA samples, and really take apart the different copy number states. So we believe that phasing is giving us the power to do that.
Very interesting. Thanks.
Any other questions?
Okay, maybe one another regarding let me check the other lineage where you are dealing with individual parts of the tumor, or as it was said on one of the slides, it was single cells. So do you actually deal with the data from single cell analysis?
Yeah, this is actually one of my main line of research for now. And, single cell nowadays means a lot of things. It's like a category that includes like several different types of sequencing technologies. And some of these technologies are really meant and can be used to infer copy number alterations. Others are meant for other different types of genomic alterations. So we are strongly working with technologies that allow us to look at copy number alterations in tumors. And my recent work, Chisel, is like the best example for it for now.
So my question would be, in those approaches, you always use whole genome amplification to actually get enough DNA to analyze when you are dealing with single cells. But all those methods bring quite heavy artifacts, especially in whole genome data, and it's not really random. So do you have some means of correction, or you have any other approach?
Yeah, this is a great point. But however, the technologies that we are working at, like during the last year, at the end of 2019, 2 new sequencing technologies have been introduced. One from 10x Genomics that is called a single cell CNV solution, and the other one is the DLP technology that actually are without a preamplification. So these are the technologies that are useful for doing copy number analysis because they sequence like thousands of cells in parallel. They don't have amplification, and they are pretty suitable for copy number analysis. They draw back clearly since you don't have amplification of DNA, the sequencing coverage that you result having is extremely low. You only cover 2% of the entire genome. But I would invite you to look at the new chisel work that we publish in Nature Biotechnology, where we deal with this issue.
Okay, any other questions? Yes, Branja, please.
So maybe I would like to ask something more technical. So in Hatchet, you do this global clustering where you, I guess, try to find preferentially clusters that appear in multiple or all samples. Do you also require that the boundaries of these segments are the same in these samples? And also, is there any balancing parameter that sort of balances how many samples you see in whether you prefer really more frequent clusters or clusters that only appear in one sample?
Yeah, so this is a very good point. So every cluster is actually composed of the same genomic regions across the same samples, but there are no constraints about these genomic regions. So the genomic regions can be like randomly distributed across the genome, not in specific boundaries. And so, on one side, what we observed is that using this approach, you have really much more power to distinguish what is variance from what are real events. But on the other way, you're not using the local information of the genome. So often, for example, you result in losing small copy number alterations, or, for example, alterations that are present in small clones, so they have only small shifts. So this global clustering will lose these events. So there is a sort of trade-off. For this reason, we believe, that this global approach is actually giving you more power to investigate the major clonal compositions. So what are the main tumor clones that are present there? But it's not answering the question about resolution. So what are the small events that are present and that are different across different samples? In order to do that, you really need to incorporate information from the locality of the genome. And so like this local segmentation will give you like a mean to do that. So it is a balance. And in terms of parameters, we don't have any parameter about the size of the clusters, but it's a probabilistic clustering based on Dirichlet. So there will be also a balance there on how much sensitivity, how many samples they have that shift, and so on.
Thank you.